In this section, we will talk about the catecholamines, adrenergic transmission, and adrenoceptors. Certain neurons can enzymatically produce L-DOPA or L-dihydroxyphenylalanine from L-tyrosine, using enzyme called as tyrosine hydroxylase, and is the parent substance of dopamine, norepinephrine, and epinephrine. Dopamine, norepinephrine, and epinephrine are the three natural catecholamines which are enzymatically synthesized. Dopamine is the final step of synthesis in neurons containing only the enzyme required for the first step, through the enzyme, the aromatic L-amino acid decarboxylase. Dopamine is used as a transmitter by dopaminergic neurons in central nervous system and by autonomic neurons that innervate the kidney. These knobs establish synaptic contact, albeit not always very dose with the effector organ. They also serve as sites of norepinephrine synthesis and storage. L-tyrosine is taken up by the endings and transformed into dopamine. In adrenergic stimulation, this step is accelerated by protein kinase A. This yields a larger dopamine supply. Dopamine is transferred to chromaffin vesicles, where it is transformed into norepinephrine. Now, how norepinephrine is released? Norepinephrine is exocytosis into the synaptic cleft after the arrival of action potentials at the nerve terminal, and the initiation of calcium influx. Norepinephrine, the end product, can be reuptake or reabsorb, which inhibits further dopamine synthesis, called as negative feedback. Four main types of adrenoceptors, which are alpha-1, alpha-2, beta-1, and beta-2, can be distinguished according to their affinity to epinephrine and norepinephrine, and to numerous agonists and antagonists. All adrenoceptors respond to epinephrine, but norepinephrine has little effect on beta-2 adrenoceptors. The activities of all adrenoceptors are mediated by G proteins. Deactivation of norepinephrine in the synaptic cleft takes place by diffusion of norepinephrine from a synaptic cleft into the blood. Secondly, extraneuronal norepinephrine uptake and subsequent intracellular degradation of norepinephrine by catecholamine O-methyltransferase and monoamine. Thirdly, active reuptake of norepinephrine, which is 70%, by the presynaptic neuron terminal. Some of the absorbed norepinephrine enters intracellular vesicles. Some is reused and some is inactivated by monoamine. So also produced negative feedback. As discussed before, stimulation of presynaptic alpha adrenoceptors autoreceptor, which inhibits the further release of norepinephrine. Now, moving into detailed discussion of receptors. Four main types of adrenoceptors, which are alpha-1, alpha-2, beta-1, and beta-2. Activation of alpha-1 adrenoceptors mediated by GQ proteins and phospholipase C-beta leads to formation of the second messenger's inositol triphosphate, which increases the cytosolic calcium concentration, which cause contraction of blood vessels, bronchiole, sphincter, and uterus, and diacylglycerol, which activates protein kinase C. GQ protein-mediated alpha-1 adrenoceptor activity also activates calcium-dependent potassium channel. The resulting potassium outflow hyperpolarizes and relaxes target smooth muscles. For example, inhibition of the gastrointestinal tract. Other functions of alpha-1 receptors are increased CNS sympathetic activity, increased glycogenolysis in liver, in kidneys, threshold for renin release rises, and its effect on smooth muscles is that it trigger contractions in the arterioles, uterus, deferent duct, bronchioles, urinary bladder, gastrointestinal sphincters, and dilator pupillae. Activation of alpha-2 adrenoceptors, mediated by GI protein, inhibit adenylate cyclase, so decrease CAMP synthesis and phosphokinase A. At the same time, increase the open probability of voltage-faded potassium channels, lead to hyperpolarization. When coupled with G0 proteins, activated alpha-2 adrenoceptors also inhibit voltage-faded calcium channels and decrease calcium influx, result in inhibition of exocytosis or decreased secretion of salivary glands, insulin, acetylcholine, and norepinephrine. Other functions of alpha-2 receptor are it decrease sympathetic activity, decrease salivation, decrease insulin secretion, decrease lipolysis, increase platelets aggregation. All beta adrenoceptors are coupled with GS protein and releases CAMP as a second messenger. CAMP then activates protein kinase A, which phosphorylates different proteins depending on the target cell type. Norepinephrine and epinephrine work through beta-1 adrenoceptors to open L-type calcium channels through CAMP and pKa in cardiac all membranes. This increases the calcium influx and therefore produces positive chronotropic, dromotropic, inotropic effect. Activated GES protein can also directly increase the open probability of voltage-faded channels in the heart. So, calcium influx increase that drives the heart, and in the kidney, the basal renin secretion is increased through beta-1 adrenoceptors. Activation of beta-2 adrenoceptors by epinephrine increases CAMP levels, thereby lowering the calcium. This dilates blood vessels, bronchiole, and uterus. Other effects of beta-2 activation are dilation of skeletal muscles and relax the muscles of the uterus, deferent duct, and gastrointestinal tract, increased insulin secretion and glycogenolysis in liver and muscle, and decreased platelet aggregation. Heat production is increased through beta-3 adrenoceptor on brown lipocytes. Now, before ending our lecture, let's name few agonists and antagonists of adrenoceptors. Phenylephrine is agonist of alpha-1 receptor, but antagonist of alpha-1 is prazosin. Clonidine is agonist of alpha-2 receptor, but antagonist of alpha-2 receptor is yohimbine. Isoproterenol is agonist of beta-1 receptor, but antagonist of beta-1 is atenolol. And salbutamol is agonist of beta-2 receptor. This was all about the autonomic nervous system. Stay tuned to sqadia.com for more precise and knowledge stuff videos.